Thank you, Robin. Welcome, everybody. Uh, share my screen here. Not totally. Yeah. All right. All right. Thank you so much for the introduction. Like Robin mentioned, my name is Saumya Ramanathan. I'm an assistant professor of biochemistry and molecular biology at Fisk University. And the title of my presentation today is Tumor-Specific Antigens, Letting Cells Go Unchecked. So first, I'd like to, before I talk about the research, I'd like to do a shout out to the university that I'm at, which is Fisk University. It is a historically Black college or university located in Nashville, Tennessee, was established in 1866 and has a very rich cultural history associated with the civil rights movement. Some of the notable alumni include St. Elmo Brady, who's the first African-American to have received a PhD in chemistry. And of course, other known alumni include the late Representative John Lewis and W.E.B. Du Bois. At Fisk, I wear multiple hats, but the research is why we're here today. So as far as research goes, I run a research lab with 11 undergraduate students and 3 graduate students. The graduate students are a part of the Fisk Vanderbilt Masters to PhD Bridge program, and I'm currently funded by the NSF for running both course-based authentic research experiences and for running my own research lab. So what do we do at Fisk? We like to think that we're cancer biologists and we study cancer at the molecular level. So before we talk about cancer at the molecular level, I want to call your attention to some cancer statistics. So I know we're all very concerned about COVID right now, but in the year 2017, 1.7 million new cases of cancers were reported, and 0.6 million people died that year of cancer. And the top 3 types of cancer that takes most lives, that is lung and bronchus across the board, prostate and breast in men and women, and colorectal cancers also have a few different things associated with them. First is that there's lots of health disparities. Second is that they have a lot of epigenetic causation associated with them, so gene-environment interaction. And third is that if they're detected early enough, then there's better patient prognosis. So I told you that I study cancer at the molecular level. So for the students in the audience, what is cancer? Cancer is a mass of cells that results from unchecked division. So you have a normal cell over here. It acquires a DNA mutation, which could be due to heredity, UV radiation, chemicals, erroneous DNA replication, smoking, or viruses. And that mutation then allows the cell to proliferate in an uncontrolled manner. The mutation usually manifests itself in 2 different types of genes, oncogenes or tumor suppressors. In general, tumor suppressors are deleted or mutated and inactivated in cancers, whereas oncogenes are amplified, overexpressed, or mutated to the point that they're always active. So at any given point in time, the balance between the tumor suppressive signal and the oncogenic signal kind of decides where your cell is in the path of cellular transformation. So as we've taught about treating cancer, there's many hallmarks to cancer cells, and these cancer therapeutics, I mean, we've over a few decades or more, we've done a great job, clinicians and scientists, to sort of try and make these inhibitors work. So for example, we have great EGFR inhibitors that prevent sustained proliferative signaling. We have telomerase inhibitors that prevent the replicative immortality or reverse the replicative immortality in cancers. We have inhibitors of VEGF signaling, which prevents angiogenesis, which is formation of new blood vessels, so that the tumors can obtain nutrition and oxygen. So we have also, aerobic glycolysis inhibitors that reverse the change in metabolism that the cancer has hijacked, and so on. And that I'm not going to talk about. But what we have noticed is that when you have a large number of individuals with the same disease that are treated with the drug, it has variable effects, because some have positive effects, some have no effect, and some have negative effect to that drug. And that makes sense, because we don't, cancer therapeutics, just like anything else, is not one size fits all. So with the advent, of course, of the ability to perform whole genome sequencing and proteomics and metabolomic analysis of blood, DNA, urine, tissue, etc., what we hope to do is move cancer therapeutics into the more personalized realm. and use of specific drugs that accounts for specific genetic backgrounds could perhaps lead to more positive effects in the majority of the diseased individuals. So with this in mind, of course, when we start thinking about personalized cancer therapy, we have to take a look at those proteins that are expressed specifically in tumors. And again, this is a huge umbrella of proteins, and it comes in different flavors. So you have those that are specifically post-translationally altered, such as MUC1 in cancers, those that are mutated in cancer, such as KRAS, of course, and those that are overexpressed, such as HER2 or c-MYC. But we're not here to discuss any of those. We're here to discuss the topic of today's webinar, which is cancer testis antigens. So cancer testis antigens are exemplified by their unique expression pattern. They are expressed in the testes during the process of spermatogenesis. Different cancer testis antigens are expressed at different time points. And then they're not expressed in any other somatic tissue, and then abnormally or aberrantly re-expressed in a wide variety of cancer cells. And in the early 90s, Thierry Boon's group discovered the first cancer testis antigen, which then they named MAGE-1, now known as MAGE-A1. And they did so by autologous typing of T cells followed by cloning. And now, currently, as of yesterday that I looked, the CT database, which is an actively curated database, consists of 220 genes that are called or can be considered cancer testis antigens. Interestingly, 50% of them are located on the X chromosome. Shown here are the three subfamilies of cancer testis antigens that my lab is interested in studying. So why hold a webinar? Why do we care? Why do we think that CTAs are interesting? So just to give you a little bit of background is that, so they were discovered as antigens. So scientists and clinicians are interested in exploiting these antigens for use as cancer vaccines. And I'm hoping that Pawan's going to talk to us about this in detail, but what we know from the literature is that their expression predicts outcomes of certain therapies. They have a demonstrated role to play in spermatogenesis, and slowly the science world has now gathered enough evidence or a body of literature that suggests that they also play a role in carcinogenesis. In addition to this, we're also interested in looking at or using them as biomarker or tools for grading of tumors. And I'll touch upon a little bit at the end of my talk here. So the overarching research goal of my lab is to understand two things. One is understand the regulation of expression of these genes and their protein products. How are they expressed aberrantly in cancers? What are the signatures that allow for their expression? And second is, what is the role they play in tumorigenesis? And the specific questions that we are seeking to answer is, are they redundant within the subfamily? Are there some that have certain signaling pathways that they modulate, whereas some others do some other functions? And kind of seeking to see why there's been recent expansion in eutherian mammals in the number of cancer testis antigens. To sort of demonstrate the kind of how we are thinking about this, I'm going to talk to you about a story on the MAGE-A subfamily that's been recently published and has been spearheaded by a graduate student in my lab, Ashley Coleman. And so what we were seeking to understand is that what are the molecular signatures that lead to expression of CTAs? So we know that in many disease states, cells go from having restrictive chromatin, where you have higher amount of epigenetic barriers and less amount of cell state transitions, to having a more permissive chromatin, where you have lesser epigenetic barriers and cell state transitions are allowed. So while of course, cancer is one such disease where your chromatin states are different. And so we were looking to see look for epigenetic markers. Specifically, we started with CpG methylation, and we're in the process of looking at histone post-translational modifications and discovering transcription factors and networks, and of course, discovering enhancers and insulators. So more on that maybe in a few years. But for now, I'm going to talk to you about the epigenetic markers that we've studied. So the first thing we looked at is we performed analysis methylation of MAGE-A promoters, and we enumerated the amount of methylation in MAGE-A promoters in a few different tissues. We did so in heart and normal colon, where CTAs are not expressed, and so MAGEs are not expressed. And we also did the same thing for testes, sperm, and in cancer, where they are expressed, with the hypothesis that we would see differential methylation when we looked at heart and normal colon versus that in testes, sperm, and in cancer. And that's exactly what we saw. which is if you looked at heart versus testes and sperm, there was a concomitant decrease in methylation. So heart is a tissue where MAGE-As are not expressed, and you see higher amount of methylation. And when you go to testes and sperm, you see lesser amounts of methylation. This was also recapitulated in the cancer setting, which is when we looked at benign, low-grade, and high-grade cancer, we see a concomitant decrease in the amount of methylation in the MAGE-A promoters as the cell becomes more cancerous. And this was really great, but we had to kind of corroborate this with experiments done with cells. So what we did is we treated cells that do not express a lot of MAGEs with azacitidine, which is a DNA methyltransferase inhibitor. And we were able to elicit expression of all MAGE-A genes almost to the same extent as we would see with patient-derived colorectal cancer cells. Our negative control here was a type 2 MAGE, MAGE-D2, which is not a cancer testis antigen. So therefore, you did not see a regulation in expression by modulating DNA methyltransferase activity. This was really encouraging to us because this suggested to us that CpG methylation is directly capable of eliciting expression of MAGE-A genes. And so then we wanted to look at whether there were other physiological states that can affect MAGE expression. And the thought for this there was precedence for this was already set in 2001, where this study actually showed that in lung bronchial epithelial cells of patients that do not yet have lung cancer, but are smokers, you can see expression of MAGE-A1, A3, and B2. So what we wanted to see was are there other physiological states? Can we catch a cell earlier, or can we treat the cell with other physiological conditions to elicit expression? We used sodium arsenide, which is a DNA damaging agent, and we saw that you do indeed see expression or elicitation of expression of several MAGE-A genes upon treatment with sodium arsenide. So this is really interesting to us because now we can move further and kind of start thinking about, okay, so now we can elicit expression of MAGEs. What is the consequence of expressing these MAGE genes? So to do that, what we did is we expressed many MAGEs. Not all of them are listed here, but this is just representative data. We expressed MAGEs in cells that do not normally express them, and we see that you can see an increase in proliferative phenotype when you express certain MAGEs, and that is recapitulated in terms of colony formation in an anchorage-independent manner compared to vector control. What we also surprisingly saw is that not all MAGE-A genes were capable of doing only proliferation. So shown here is MAGE-A5 and A10, two very closely related MAGE-A proteins that you can see is when you treat with high doses of a chemotherapeutic agent, such as 5-fluorouracil, is capable of conferring resistance to cell death, and actually cells are viable in the presence of this chemotherapeutic agent compared to control cells. So this is really interesting to us, and this was really exciting because this suggests to us that not all MAGE-A genes are redundant, not all have a proliferative phenotype, that there may be other signaling pathways that we could follow. So to kind of summarize this really short story that I talked to you about is that we were looking for how the MAGE-A genes are expressed. We were using them as exemplary members of cancer testis antigens, and we are hoping to expand our studies to other cancer testis antigens as well. We've found that DNA methylation does indeed directly regulate MAGE expression. We've also found that certain MAGE-A genes do increase cell proliferation and anchorage-independent growth, but some other MAGE-A genes cause drug resistance. And eventually what we're hoping to put together as a puzzle is that what is the role that these genes are playing in germline and in cancer? And there's some evidence in the literature that suggests, of course, that these genes are protecting cells, whether in germline or in cancer, from extraneous stressors and allowing for survival. So with that, I'm going to take a few moments to acknowledge all the hard work of all my students. Ashley is currently at Duke. She's doing her PhD at Duke. Angel was a Mark Scholar in the lab who's doing her PhD at Cincinnati. Jodine is currently doing her PhD at MCB in Seattle. And Sari is actually headed to graduate school next year and is interviewing. So if you come across her, give her an admission. Shayla is in law school, and she also spearheaded the MAGE-B project, which I haven't had time to talk about. I would like to thank NSF for funding, the Bridge program for funding, ASBMB, a huge shout out to Robin, who's kind of taken this webinar from conception all the way to today, and Laurie for today assisting us, and all the cores at Vanderbilt, because that allows me to get my research done. And with that, I'm going to end my show and turn it over to Brian. We'll take questions all at the end, which will hopefully facilitate a lot of discussion. Stop share.
Great. Good. Let me go ahead and share my screen here and get started. So, so just brief introduction. I'm Brian Ladle. I'm a pediatric oncologist and translational cancer researcher, so physician scientist at Johns Hopkins University. And I specifically am interested in how we can employ immune therapy efforts for pediatric solid tumors, specifically pediatric sarcomas. And so Saumya reached out, it's been several months ago now, wondering if, what my interest would be in presenting at this seminar. And, I jumped at the chance, and I'm excited to be able to share some of my thoughts about cancer testis antigens, obviously not my thoughts, but in the field as well. So Saumya gave a great, kind of overview of cancer testis antigens. I liked her slide, why are they interesting? And I think each of our talks today, will maybe highlight a couple of reasons of why they're interesting. But what I wanted to do, I wanted to talk about them specifically from the lens of an immunologist, So I'm an immunologist, and so just want to talk about that. So I'll start with this picture. So I'm not just an immunologist, but a tumor immunologist. And what I have here, these are obviously some IHC slides, and this is looking at this tumor here, happens to be called synovial sarcoma. And what you see is that here's two samples from the literature of immunohistochemistry staining of these tumors. And you can see this beautiful differentiation between normal tissue and tumor tissue. And here's another example here. And what is more interesting as an immunologist is that you see high expression, you see uniform expression, in just the cancer cells and not the tumor cells. So, of course, those who would know, this is looking at NY-ESO expression as one of the, most famous cancer testis antigens, and was specifically highlighted and discovered because of its potential of being a cancer-specific antigen, something where the immune system could localize and destroy only cancer and leave normal tissues alone. So thinking of that lens, so as an immunologist, what would be an ideal immune tumor target? So I want to maybe think through this and how well do CTAs fit this definition? So I guess my recipe for an ideal tumor target would be one that you'd have high uniform expression on every cancer cell. Second, that you would have no expression at all in normal tissues, and that if you can generate a great response to this antigen, you would hope that it would leave your normal tissues completely alone. Third, that it has to be recognized well by immune cells, by T cells. I realize I'm leaving out other immune cells, but for today's talk, so many minutes, we'll focus on T cells. Third, that because it's a T cell target, you have to have persistent presentation by MHC class 1, so that T cells can actually see that target that's present in tumor cells. And then lastly, maybe an ideal would be that the tumor is dependent on expression of that antigen for survival. And that, if the immune system gets rid of that target, will the tumor also go away, as opposed to just becoming just escaping with an immune or with an antigen negative, selection process. So let's think about each of these and how would CTAs fit as far as this definition goes. So first, our goal of having high uniform expression in every cancer cell. So CTAs, there's various of them. This is a recent paper just looking at three, PRAME, NY-ESO, and SSX2. And this is looking at a variety of, these are soft tissue sarcomas specifically. And you can see that not all soft tissue sarcomas express these at high levels. Here's PRAME, NY-ESO, and again, this is, some tumors do, express very high levels, but others are rather heterogeneous, and some even don't express it at all. they quantitated this looking in their data set, right? So how many of these tumors that they looked at expressed high levels of these antigens? And you can see that, right, it varies between the diagnosis, and this is again just a sampling of sarcomas, and but you can see that some have, you know, I guess a significant percentage of them express high levels. So one of the most commonly targeted is NY-ESO-1 and synovial sarcoma, but you can see only about 40–50% of tumors will actually express high levels of this. Lung cancer is another diagnosis where people try and target these cancer testis antigens. Again, this is another paper where they, the goal is looking how prevalent is expression of these antigens in lung cancer. You can see that some expressed very little, up to some expressed a lot, and then quantitating it, looking at the samples that they did, right? You see, for instance, gauge and NY-ESO-1, in this case, only 20, maybe 10% of these tumor cells expressed high levels. So as far as CTAs being an ideal tumor target, right? I'm going to give this a plus and a minus. That there are some tumors that do meet this criteria, that really do have high expression in every cancer cell, but there certainly are some that don't. And for each diagnosis, even in that diagnosis, there's variety there. So let's talk about that goal of, right, that you would not have any expression in normal tissues. How well do CTAs fit this goal? So this is just RNA-seq data taken from PubMed gene database, and this is looking at NY-ESO-1, or the gene name is CTAG1B. And you can see this is the type of data that gives these genes their definition of cancer testis antigens. That in normal tissue, right, expression levels, again, are very, very low, but there is significant expression in testes. You can see that there may be low expression in other tissues. For instance, here's placenta, maybe a little expression in the heart. And so even though there is high expression in testes and in cancer, right, you worry, could there be on target, meaning your immune system's hitting the target, but off tissue effect, or I guess off cancer effect. So here's looking at IHC, right? That other was RNA data. So you see in testes that, for instance, this is MAGE-A4. You do see expression in some normal tissue in placenta. Again, here's highlighting some of the heterogeneity of CTA expression. This is in a bladder carcinoma. So even though you perhaps have very low expression in normal tissues, is some expression a problem? And this is something that the field is grappling with as we're able to really generate potent immune responses to some of these antigens. This is a published example targeting the CTA MAGE-A3, and these were using T cells that were known to be specifically targeted to this antigen. This is a clinical trial in patients, several of which had very good responses in their tumor, but also some of these patients developed severe neurotoxicity and even death from some of them. And this is just highlighting the MRI scans showing the severe inflammation and enhancement that occurred in some of these patients that received these T cells that, again, were specific to this cancer testis antigen, but did have some on-target but off-tissue effect. So again, it just highlights that are these ideal as far as no expression in normal tissues? Plus and minus. Some of these CTAs that are targeted have not seen other effects like that, but some have, and it's something we have to be careful with as we generate potent immune responses. So next, an ideal immune target will obviously be recognized by T cells, and importantly, the definition is yes. This is how these genes were actually identified. Work with Terry Boone and Lloyd Old. Right, some of the initial labs, how they found these antigens were actually looking at immune responses in cancer patients. But you have to then think of, all right, how is a T cell going to recognize these antigens? Most of these proteins are cytoplasmic or nuclear proteins, meaning they're intracellular versus a membrane-bound protein, whereas membranes more accessible to the immune system and perhaps can be targeted easier. Whereas these cytoplasmic and nuclear proteins, in order for the immune system to see them, they have to be processed and presented on the MHC molecules. Antigen-presenting cells have to be able to take them up and present them. So as such, they have MHC restriction, meaning they are only presented by certain MHC molecules, and this has limited some of how we can target these antigens therapeutically. So just briefly overview of some of the therapeutic approaches to generate T cells against these antigens. One is just growing and expanding tumor infiltrating lymphocytes in vitro and then reinfusing them into the patients. This is work pioneered by Steve Rosenberg and group at the surgery branch at the NCI, as well as others. Other approaches include vaccination, meaning, all right, if we know these are relevant antigens, let's just vaccinate, mount an immune response against them, and multiple efforts have been made there. I'm just going to highlight a little more this kind of new recent approach, which I think is getting more traction as far as using engineered T cells that express T cell receptors specific to the cancer testis antigens. And this is done by cell engineering to introduce in these T cell receptor sequences. So one of those promising constructs so far has been developed by Adaptimmune and now GlaxoSmithKline. It was just given FDA breakthrough approval for treatment of synovial sarcoma, and second and third generations of these engineered T cells are coming into clinical trial. So how these work is these are engineered autologous T cells. So you take the T cells from the patient, you add in with retrovirus or lentiviral vectors to express these specific T cell receptors that we know are specific to these antigens, and in this case specifically NY-ESO-1. These T cell receptors have been altered slightly to have even higher affinity for their specific epitopes. And at least in synovial sarcoma, you recall that was the first slide I showed, where synovial sarcoma, many of them have very high, very uniform expression of NY-ESO. And in these patients, they've seen an over a 50% overall response rate with these engineered T cells. So certainly some exciting data and more to come there. So as far as recognized by T cells, yes, absolutely they can be. What about persistent presentation by MHC class 1? So this is certainly an issue with immune evasion, and we could give a whole talk about mechanisms of immune evasion. But some of the common mechanisms are tumor cells are able to downregulate MHC class 1 and other antigen processing machinery, so that even though you might have a mutated or CTA expressed in the tumor, the immune system is not going to see it. And then other specific concern is antigen loss, and that when you mount a great immune response against an antigen, the tumor potentially can just lose expression of that antigen and keep growing despite a great immune response. And so this is specifically a concern when you're targeting, for instance, one single antigen. And then finally, right, would the tumor be dependent on its expression for survival? And this, I think CTAs don't necessarily fit this very well, right? We're learning more of what their role is in tumors as far as, as Saumya mentioned, growth and things. But as far as being the driver of these tumors, we don't typically think of CTAs in this way. So I'll just spend the last couple of minutes talking about some of the work that we're doing in our lab. And specifically, can we broaden the use of cancer testis antigens as targets, right? We've shown that some tumors express a lot of it, but could we have methods of increasing and making a CTA a target in a tumor that might not express it normally or express it at low levels? For me, this is especially interesting as I'm interested in pediatric sarcomas, which have very low tumor mutation burdens and few immune targets. And so one question that we've had is that can CTA expression be upregulated using epigenetic modifying drugs, right? As Saumya mentioned, we know that these are controlled by methylation. So can we increase their expression and potential antigenicity using epigenetic modifying drugs? We use two sarcoma models in our lab, a KP-SARC model, which is a KRAS P53 driven, and a rhabdomyosarcoma line. These lines are locally aggressive, very poorly immunogenic, have low mutation burdens, potentially a good model to look at this. So the two classes of drugs we've used, as Saumya mentioned already, DNA methyltransferase inhibitors that potentially will release or cause demethylation of these hypermethylated promoters and allow expression, and then histone deacetylase inhibitors. So looking at a panel of cancer testis antigens, these are mouse cancer testis antigens, and in these sarcoma cell lines, we do see, as Saumya mentioned, treating with DNA methyltransferase inhibitors gives significant induction of these genes which are normally silenced in untreated cells. HDAC inhibitors by themselves don't do very much for cancer testis antigen upregulation because of the methylation. But the combination of doing DNA methyltransferase inhibitor followed by HDAC inhibition actually increased expression of these even further. So in the lab, we specifically wondered, all right, we can upregulate the expression of these antigens, but can we then generate a good immune response against these that can control tumor growth? So we treated these cells ex vivo, right, in vitro, and then injected them into mice. So these are our epigenetically modified tumor cells now, and looked to see do they grow differently. And importantly, untreated compared to decitabine and entinostat, those are the epigenetic inhibitors. Just treating the cells by themselves, the cell fitness and ability to grow in the mice did not change. But what we wondered is, can we generate an active immune response against these newly expressed antigens that could control tumor growth? So we use a cancer vaccine approach. We term it GVAX, where we use tumor cells and mix them with a cell line that produces GM-CSF, and that becomes a whole cell cancer vaccine. And what we did was we vaccinated mice either with GVAX with untreated sarcoma cells or with epigenetically primed sarcoma cells that potentially express all these new antigens. We vaccinate the mice, give them time to mount an immune response, and then challenge them either with untreated tumor cells or with these epigenetically primed tumor cells, and see if the immune system is now able to control growth based on these epigenetically modified proteins. So just showing just a snippet of the data. For instance, when we give this vaccine, the epi-GVAX approach, and give untreated tumor cells, there's really no control of tumor growth. But if we challenge with tumor cells that have been epigenetically primed, right, potentially express these antigens, we do see significant delay of tumor growth. And if we add an immune checkpoint therapy, anti-PD-1, anti-CTLA-4 in these mice, they're able to completely control and eradicate the tumor growth. We have shown that this is a T cell dependent process, and that if we eradicate CD4 and CD8 T cells from these mice, these tumors grow normally, and right, that pre-vaccination does not preclude tumor growth without their T cells. So again, this is just a snippet of things that we're interested in, and glad to talk about more during the discussion. But in conclusion, right, as an immunologist, how good are CTAs as an ideal immune target? They really are quite good in the right setting, but they certainly have limitations, and those are things that some of the things we've talked about. And next would be, can we use epigenetic modifying drugs such as DNA methyltransferase inhibitors or HDAC inhibitors that could potentially broaden our ability to target CTAs, and specifically in tumors that might not express other antigens or have low CTA expression to begin with. And these agents also have limitations, and you know, which we could discuss later. So yeah, I just want to thank the organization today, right, for hosting this talk. I want to thank members of my lab who have done all this work, our collaborators, as well as our funding organizations. And so thank you. I appreciate the time, and we'll take questions at the end.
Thank you, Brian. I apologize. I was supposed to introduce you, but you did a great job of introducing yourself. So I'm going to, as we turn the screen over to Pavan, I'm going to introduce Pavan, since I was supposed to do that. Dr. Bashi Reddy got his bachelor's at Harvard University, and he got an MD at Stanford. And then he did a residency and a postdoctoral fellowship at Dana-Farber and Brigham Women's Hospital. He is currently transitioning, or recently transitioned, as he just told us, as of last Friday, to his newly minted assistant professorship at MD Anderson. And so just a shout out is that he's looking to hire y'all, so make sure that you get his contact information. And over to you, Pavan. Thank you so much for being a panelist, and thank you, Brian, as well.
Great. So yeah, so good morning or good afternoon, depending you know, where you are. And first of all, just like Brian and Saumya said, you know, really appreciate the American Society for, you know, Biochemistry and Molecular Biology, especially Robin, for, you know, organizing this along with Dr. Ramanathan. And so I think, so I'm obviously honored to be here. I'm really excited. I think it will be, I think hopefully the audience, you'll get a good view of what Dr. Ramanathan showed, which is how, in the center of this cancer testes antigen universe, all the different aspects of biology that intersect with it and all the different ways we can get there. And so I think hopefully what I want to share with you today is a story where we actually set out not actually to study cancer germline antigens at all, but ended up there using an unbiased discovery approach, which I think hopefully highlights actually some of the essentiality of the biology that these genes and proteins can play, certainly within cancer immunotherapy, and potentially in a different way, complementary to what Brian had just spoken about. So this is basically a visual abstract of kind of the talk, and you can see the paper below. This is in close collaboration with Dr. Shukla at the Dana-Farber. And so what my lab focuses on is trying to understand why patients do and don't respond to immunotherapy. So I'm an oncologist, and patients that come into our office, what we would love to do is take biopsies and interrogate those biopsies, because we believe that that's sort of where the activity and where the information is from those tumors, and then try to understand, okay, which of these patients should be given immunotherapy A and which should be given immunotherapy B. And so to do this, of course, we need to identify those biomarkers that can stratify our patients. And we would love to know in which cases are those biomarkers also serving as mechanisms and providing mechanistic insights into the molecular and cellular pathways that drive responses to immunotherapy. And so this slide really is to just to show you how immunotherapy has kind of taken over our field as oncologists, especially in the adult world. And so here you can see, over at least until 2017, the increase in yearly FDA approvals for immunotherapies and the dramatic growth that's really been driven by this specific class of immunotherapies called checkpoint inhibitors. So what are checkpoint inhibitors? And so this slide is really made to highlight the two major distinct pathways for the current FDA-approved checkpoint inhibitors. And in this case, one is the PD-1, the CTLA-4 pathways. These are co-inhibitory receptors present typically on T cells, other immune cells as well. And what these molecules do is they're upregulated during T cell activation, and then when during sustained expression can actually lead to downregulation of T cell activity, specifically anti-tumor T cell activity. And so what's important to note is that that antibodies that we use interrupt these pathways and reinvigorate T cells, but they reinvigorate T cells in very different ways. So these two molecules, PD-1 and CTLA-4, occupy distinct biological niches, and this will be important later on. But despite this sort of biological understanding that we've had, we still don't know what determines response and resistance to immunotherapy. And so to do so, what we did was turn to a setting where responses are well characterized and where long-term data exist. Here you can see in this Kaplan-Meier curve, these are patients with metastatic melanoma. And you can appreciate the tail, what we call the tail on this curve. So roughly 10 to 15, even up to 20% of patients treated with CTLA-4 blockade can actually have long-term survival in what used to be an otherwise uniformly fatal disease. So immunotherapy has really transformed melanoma, which is a great test case, base case for cancer immunotherapy. So we set out then to, given this remarkable effectiveness of CTLA-4 blockade, we set out to try to understand the mechanisms of response and resistance. And specifically, we wanted to take advantage of gene expression analysis, which has the potential to yield useful mechanistic insights in an unbiased fashion. And specifically, we wanted to identify signatures that were specific to CTLA-4, but not PD-1 blockade. And the idea here was that this would enable us as clinicians to effectively pair the right patients with the right treatments, as well as potentially establish a framework for rational combination therapies. And so the question that motivated this work was, are there transcriptomic signatures and mechanisms that were unique to one, CTLA-4, but not PD-1 blockade? And so I'll take you through the story where we first will look at what are the transcriptomic data sets that we used. And hopefully you'll also get an appreciation
All right. Pavan, I think we lost you.
Yeah, it looks like we did. Saumya, this is Lori.
Okay, so why
Okay.
don't we take, why don't Brian and I take a couple of questions while we're waiting for Pavan to get back online. So Brian, the first question we have here, can you hear me, Brian? Oh yeah, okay, sorry. First question we have here is, why do you think cancer cells have chosen CTAs as their antigen marker? And what do you think are the positive and negatives on behalf of cancer cells?
Sure, right. I mean, kind of an existential question of cancer cells, right? I think it depends on the tumor. For instance, synovial sarcoma, its main genetic aberration is in epigenetic control, right? And so I think re-expression of these cancer testis antigens just occurs because of the actual driver mutation in synovial sarcoma. Right, other cancers, we know that just hypomethylation in general is a pervasive, kind of phenotype of cancer, and so I think because of that and how they're regulated by methylation, right, those antigens are just upregulated. And then, right, the biology of these, I think, is the other question, and I'm certainly not the expert in that as far as the biology of CTAs, right, and how I'm sure they lend to to the tumorigenic effect.
Yeah, so I can speak to that a little bit. So yeah, so I think Kirti, that's a really good question, because I think I mentioned very briefly also that in eutherian mammals, there's been a recent expansion in the number of cancer testis antigens. So if you look at, for example, the XAGEs, which we study, there's in chimpanzee, there's maybe one XAGE gene, but in humans, there's 4 XAGE genes. So clearly there's some sort of evolution going on that expands the number of these genes. So now we don't know whether the expansion is because of the role they might play in reproduction, which would certainly make sense in terms of evolution, survival of the fittest, and so on. But it also might make sense in terms of cancers as to how many different kinds of CTAs can cancers express, and sort of thinking about coevolution of the MHC class molecules as well. So it's kind of a very existential question. And we think that there are certain CTAs that definitely give cancers a proliferative advantage. And that might be triggered by epigenetic memory of them giving advantage during reproduction, if that makes sense. So I hope we answered your question, but as to why they've chosen them as antigen markers, I think it's because there's many different kinds of CTAs, so you could perhaps think about how many different kinds of antigens to present. So I think that's kind of where the field is headed. So the next question is, did you, so I think this is for you, Brian, did you also look at the immunopeptidome after the treatment?
Right, yeah, that work we're ongoing, right? One of the issues with a whole cell vaccine approach is that you don't know what the relevant antigen is, right? It could be a CTA, it could be something else. But we've chosen this approach, and now kind of looking backwards, right? What immune responses are we generating? Because I do feel pretty strongly you need a polyclonal response, right? You can't just target one antigen. And so, right, hopefully next year I'll have a better answer to that question.
Sounds good. Okay, Ames was asking to me, what cell line did you use for proliferation viability? I believe the data that I showed you was regular hex cells, but we've done the assays in human colonic epithelial cells that are immortalized but not transformed. We've done it in human mammalian epithelial cells, HMECs, and we've done it in HBECs, which is bronchial epithelial cells as well. And so the consistency is pretty across the board, pretty good. So I hope I answered that question. And then the next question is, Indu asks, as cancer can be considered as a lifestyle disease also, is there any effect of diet on expression of CTAs? That's a great question. So we're kind of trying to figure that out. I think we're trying to figure out all the physiological states in which we can elicit cancer CTA expression.
There we go.
There we go. Yay, Pavan, you're
Perfect.
back.
Great.
Okay, we're going to come back to Indu's question as soon as we get Pavan up and running.
Yeah. Okay. Sorry about that. Maybe MD Anderson is having second thoughts about hiring me, but my access is, my Wi-Fi is back going now.
It's too soon. It's too soon.
It's too soon. So I'll go back a few slides and apologize. So I think maybe you saw this, where I just sort of highlighted the point was that there's a specific set of MAGE genes that we found to be upregulated. So not all of them, but a specific subset of MAGE genes, and that's important because the MAGE genes are divided into 4 families, and this family is actually autonomously regulated from all the others. So it seemed like there was an underlying biology here. We called them the CRMA genes just to make it simpler, and we also made sure that, importantly, because we found this in a discovery cohort, that we also could see this in a validation cohort as well. And so we looked into another cohort, and we can see again these same MAGE genes. The same subset of MAGE genes are enriched in these, in this case, progressive disease patients, so patients who didn't respond to CTLA-4 blockade. So this was at the RNA level. What about at the protein level? Here we used a MAGE antibody. Now it's not specific to this subfamily, but it recognizes multiple MAGE family members. Nevertheless, you can still appreciate the enrichment of MAGE positivity in these no benefit groups. You can see an example staining here, again showing that both baseline transcript and protein levels are clearly important for correlating benefit, or in this case, primary resistance to CTLA-4 blockade. So why is this? And are these genes really just a biomarker, or is there actually a distinct biology that's happening here? So to understand this, again, first we looked into our transcriptional data by going to the Cancer Genome Atlas. And here, our hypothesis was that, well, if we divided the metastatic melanomas in TCGA by those that are high expressing for these CRMA genes versus low, and we perform a differential gene expression analysis, that presumably the genes associated with high CRMA expression should also significantly overlap with genes that associate with these no benefit samples. Because remember, we had bulk RNA sequencing data associated with them. And in fact, we did see a significant overlap between genes associated with the CRMA genes in TCGA, as well as those associated with this primary resistance phenotype, suggesting that in fact there is an inherent CRMA-associated transcriptional program that's driving primary resistance. So what about how are these genes regulated? And Saumya and obviously Brian got into this a bit. And so we performed methylation-specific, locus-specific methylation analysis, really looking at the promoters from MAGE3 and MAGE6. And again, you can appreciate the increased methylation at this locus, at the promoters in the clinical benefit group compared to the no benefit group. And this makes sense as to how these genes are being regulated in these patients, but it ties that promoter methylation to clinical outcomes. And then to get into the basic biology and we wanted to see whether there were genome-wide epigenetic effects of this MAGE upregulation. So now what we did was we went back to TCGA. We did a differential methylome analysis between the tumors with high CRMA expression and those with low CRMA expression. And now what you can see is this predominance on this volcano plot, this asymmetry of methylation probes across the genome in these CRMA low tumors, meaning you're seeing a global hypomethylation event in these tumors that upregulate CRMA. So potentially there's a wave of demethylation that happens, and that is also hitting the CRMA genes. And you can also identify some of the pathways that are implicated by this methylation. And it turns out these are the same pathways that we see in our clinical discovery cohort as well. So clinically, we want to know, is the CRMA signature actually predictive, or is it simply just prognostic and it's associating with a more aggressive biology? So this is important because first we had already shown that the CRMA expression was associated with response, and we can also see that it's actually associated with survival too. So the patients with low CRMA expression survive longer in our discovery cohort than those with high CRMA expression after treatment with CTLA-4 blockade. And importantly, we can do a multivariable analysis. And for some of the immunologists out there, some previous predictors that have been shown, like the number of mutations that you have to associate with outcome, actually are far less predictive than the CRMA expression. So when you bring this in, this is the only significant variable in this multivariate analysis in our cohort. So this is great. It suggests, of course, CRMA expression predisposes to poor survival after CTLA-4 blockade. But again, is it predictive? And so when you go to TCGA and you look at all the patients with melanoma and you divide them into high and low CRMA expression, you actually don't see any difference in survival. And these patients weren't treated with ipilimumab. So now you can see that the CRMA expression is actually not really prognostic in this setting, but it really does seem to be predictive and tied to the mechanism of action of CTLA-4 blockade. And finally, we wanted to make sure that because CTLA-4 and PD-1 occupy distinct niches, we should see that this doesn't, that this is not specific to PD-1 blockade. And as you can see, there's a lack of any differential regulation in either of two cohorts treated with anti-PD-1 antibodies. So really suggesting what we're finding, these MAGE genes are doing something unique and specific to the blockade of the CTLA-4 pathway. So how? What are they doing and how are they doing it? So, and Brian touched on this a little bit, for a long time, many folks have thought of the cancer testis antigens as not really having anything to do with driving oncogenesis, but actually as immunotherapeutic targets. But recently been a lot of data specifically from Dr. Ramanathan's previous mentor, Ryan Potts, where you can see that MAGE genes associate with and can drive tumor properties such as sustained proliferative signaling and invasion. And some of how they do this is because these MAGE genes bind E3 ubiquitin ligases, and importantly, they can suppress autophagy. And this is important to recognize because autophagy is actually important for exciting and alerting the immune system to attack tumor cells and to attack cells that die through this process of immunogenic cell death. So I'm gonna skip this a little bit. This is a complicated slide of the immune cycle just to show you that autophagy and immunogenic cell death occur far upstream of where the CTLA-4 pathway acts. And so our hypothesis was that autophagy would be suppressed in human melanomas with elevated MAGE protein levels. And I'll go through this a little quickly. And so these were three approaches that we took. And so firstly, what we showed was that in fact, if we look at other protein databases, we can see other proteins related to autophagy that are indeed suppressed by tumors with high CRMA expression. And we found a critical protein involved in autophagy to also be targeted by the MAGE-A TRIM28 complex. And this is in association with Ryan Potts, a previous mentor from Dr. Ramanathan.
Did we lose him again?
Yes, I think we did. Again, I will call him and get him back online.
All right, so I think we can answer Brian. I think we were about the lifestyle disease. So I think that we were trying to look at it, but I don't know that there's a lot of data that that can back up, I guess, the claim that diet express causes changes in expression of CTAs. I think this is a great question. Induka follows up with her question to Brian, is that of the cancers of the same type, is there a difference in of CTAs in pediatric and adult cancers, or any CTAs more specific for, are any CTAs more specific for pediatric cancers?
Yeah, that's a good question, and I'm not sure I know all the specifically the answers to it. Some pediatric cancers actually do not express them because, some of these pediatric cancers, they're translocation driven. They're just very specific pathways, and it doesn't really mess with the methylation per se. Others, for instance, like I mentioned, synovial sarcoma, it's part of the biology of the cancer driver, right, that upregulates it. So it's a good question. I just don't know all the specifics.
Yeah, and I don't think that I've analyzed actually the difference in expression between pediatric and adult cancer. So that's might be something that we could look at. I'm sure we could look at that. Okay, so the next question we have is, do CTs show a larger copy number in gene copy or levels of expression in more invasive cancers? So I think I can briefly answer that. I have not seen cancer testis antigen to be I haven't seen a copy number change in any cancer testis antigen in any of the tumors that I've analyzed with the TCGA and some sequencing that we've done. So I don't know the answer to the question about whether it's more invasive cancers. But I kind of, we do hypothesize, kind of going offshoot of that question, is we do hypothesize that within a tumor, we do think that CTs are expressed in some cells more than in other cells, and that there's some heterogeneity. And I think that Brian also talked about that. Did that Brian, do you think that you could, what do you think, I guess?
Right, yeah, I don't know a role of gene copy number and things, but right, but heterogeneity is a real issue, I guess, with targeting CTAs, right? And
Right.
Some are high and some are low, and in the same tumor, right, they'll be different.
Right. And then the last question we have is, are MAGEs regulated by P53? Off the top of my head, no, I don't. I think that regardless of P53 status, we've seen expression of certain CTAs. So I'm not sure that I can confidently say that MAGEs are regulated by P53. Yeah. So okay, since the question, we're still answering questions, I had a question for you, Brian. So do you think that I guess, is there a way that we could look at, maybe this is setting up a future collaboration, but look at antigenicity of the so many different kinds of CTs? Is there because where I'm coming from is that certain kinds of cancers, so for example, certain kinds of, so lung cancer, for example, expresses a whole bunch of MAGE genes. So maybe it's harder to find, and since they're so close in sequence, maybe it's harder to find a good antigen there. But maybe certain other kinds of cancers like glioblastoma, you see expression of only one MAGE gene. And so I think, is there any, first two parts, is there any use in parsing out the different kinds of cancers and looking much closely at a larger data set as to which cancer testis antigens are highly expressed, and then take that data set and then couple that with analysis of like epitope prediction or antigenicity prediction, and kind of do that.
Right, yeah, and I think that's why Pavan's data is so great, and we should let you finish your talk, but right,
I know.
where really looking at, all right, there are some that are predictive of immune responses, right, and there's some that are not. And the solid tumor world has the similar stories. Some diagnoses, high expression of PRAME is good survival. Other diagnoses, low expression of PRAME is good survival. Right. So it's so I think it's hard to tease out. I mean, one thing that I always think about, the tumor is expressing it and the tumor's growing. Right. So it didn't work. Right. Even if you had a lot of them, the immune system, right, still didn't work. So that's, you know, so I think just by themselves, right, you've got to have more like an active, you know, immunologic activation. Then of course checkpoint inhibitors, right, and all of those things I think come into play there.
All right, you're good to go, Pavan.
Oh, good. I was just going to add on. What I'll do is I'll answer that question, because I think that's a great question, and I will. I think the main point is this. You know, is this last slide that I wanted to show, which was the model that we had, which is we think these MAGE genes are, especially these specific ones, ubiquitinate and kind of degrade all these autophagy activators. They suppress autophagy. And we think that's important because that's driving CTLA-4 resistance. And so of these, I would say that, you know, the main point is that, you know, we can just, using this unbiased discovery approach, we see these MAGE genes are important. It's uniquely to CTLA-4, not PD-1, and I can explain why in a bit. And it seems to implicate autophagy. Now, and of course, the most important slide, this was actually almost a multinational collaboration with a lot of different groups. And of course, I am recruiting postdocs, and please email me, and I promise you the Wi-Fi will work when you get here. But but I think an important point, getting to what Brian was kind of alluding to, is that, you know, I think I think each of these cancer testis antigen families probably have very different roles, and specifically the ones that we looked at, certainly by inhibiting autophagy, I think prevent an immune response from even getting started. And you know, some of you may remember there were many trials of MAGE vaccines, and these actually failed sort of miserably. And so I think some of the reason why they failed was because the patients with the high MAGE-A3 expression were the ones where autophagy was disrupted, where the T cells couldn't be primed. And so even if those antigens were being expressed on HLA, T cells couldn't get in there because there were no danger signals to alert them. So that's, you know, one hypothesis for why those vaccines potentially failed. But understanding that immunologic link may be helpful for understanding why different antigens stimulate immune response, and some do and some don't. Yeah. But I don't know if we can keep going, but if I can squeeze a question to both of you, one thing I thought was interesting, you know, both of you, of course, are very interested in how in the regulation of these genes, especially through methylation and using methylation inhibitors. So I'm curious if you had a sense of whether this was irreversible or reversible. So when you inhibit DNA methyltransferase inhibitors and you downregulate the methylation and allow these MAGE genes to be expressed, or cancer testis antigens to be expressed, can you take away, do you need to, is the cell now is that locus irreversibly hypomethylated? And for patients, you know, do we have to keep them on these methyltransferase inhibitors? Or do you think just, you know, one shot is enough to upregulate these antigens?
So, so experimentally, I don't think it's irreversibly methylated or differentially methylated. You can certainly bring it back to the non-expressed status, but also the key part there is that methylation is not the only marker. So there are certain histone post-translation markers that we've seen, perhaps not at the MAGE promoters, but we're in the process of analyzing some pro-seq data for enhancer discovery. And so we think that there's obviously some transcription factors are regulating expression via enhancers and not necessarily just the promoters. So I don't think it's a reversible process. I think it's very much in a flux, which kind of makes it more interesting to see exactly when, what are the conditions which you can turn the expression on, and what are the conditions when you can turn the expression off.
Yeah, I mean, when you're looking at methylation, I mean, one nice thing is that is a heritable, right, trait, and that as that cell divides, it will continue to be hypomethylated. So at least in our studies, and others have shown this, right, you treat for 72 hours, you check 2 weeks later, and they're still being expressed.
I see.
You treat ex vivo, inject them, and pull out the tumors, and they'll still be expressed. So at least clinically, people have tried to do kind of pulses, right? And one of the issues with these drugs is they are somewhat chemotherapy agents, right? And so high doses will actually ablate your immune cells. So people are trying kind of low dose, you know, like a week on, a week off, you know, that sort of, you know, potentially with that, you can get the hypomethylation, but also preserve, you know, kind of normal immune function. And so that, yeah, and there's a lot of practical issues with those drugs as far as, you know, how long they last and, you know, their metabolism and things, but yeah.
Makes sense.
I think so when you use these inhibitors, you turn on the expression, and clearly we're in the camp that believes that CTAs are not passenger genes, they're actually doing something. So I guess how are we going to reconcile getting their expression up and, you know, using them as an antigen, but then also kind of offsetting that effect by preventing them to like do their thing and cause proliferation or whatever other signaling pathways they're doing. So that's kind of they've been trying to reconcile that for a few years now, actually.
Yeah. I think that will be important. I don't know if either of you know if, it'll be important to have continued work into the underlying, the cell autonomous roles that
Right.
these different cancer testes antigens have. I mean, the non-autonomous roles have been there since obviously Terry Boone, discovered these in 1990. Although there's still much more to do, but given the essentiality, at least of the MAGE 3 and 6, those great papers from your previous lab, I wonder how much that essentiality also translates to other cancer testes antigens, and is this oncogene addiction model applicable to all of them or just a subset of them?
Right.
Yeah.
So we have 2 more questions. I think we should take them since we lost Pavan, and this question is for Pavan. Why is MAGE A family not a player in PD-1 blockade?
Yes, great question. And I probably set up for that. So the answer is probably because autophagy is really important for T cell priming, and that's restrained by the CTLA-4 pathway. And so that makes sense why autophagy suppression might impair priming and therefore and exists all upstream of the CTLA-4 pathway. PD-1, on the other hand, acts in the tumor microenvironment. So PD-1 is basically, the PD-1, PD-L1 interaction is basically the stop sign or the shield tumors put up right when T cells are juxtaposed and are physically adjacent or binding and even recognizing the antigens that tumors express. So probably what we think is happening is that the patients who are responding to PD-1 blockade, the T cells are already there. They're already in the microenvironment, and a lot of them already recognize tumor antigens. So suppressing autophagy is not really going to help the tumor that much in terms of evading PD-1. Whereas for CTLA-4, it definitely will. And then one secondary answer is that PD-1, the expression of PD-1 is also very promiscuous, whereas CTLA-4 is pretty much only on T cells and maybe B cells, but people have reported PD-1 even on melanoma cells. So I think that makes it complicated to find those signatures in that setting.
All right, we have time for one more question. This is to all panelists. Are CTAs lesser expressed in female reproductive system? Also, is there a difference in CTA expression in the same cancer, same type of cancer, from males and females? I don't know. I cannot answer the second part of the question because when you analyze TCGA database, they're kind of de-identified. So we have no way of telling what sources are male or female, but is it expressed less in the female reproductive system? I think it's safe to assume that it is. But do some CTAs are expressed in ovary and uterus and cervix? So I think it depends on the CTA you're looking at.
At least in tumors, when we looked at our data, gender didn't actually have a role to play, and the tumor expression of the MAGE genes didn't differ by gender.
All right, so I think that's pretty much it. I think we've answered all the questions, which is amazing. With that, I would like to thank ASBMB, Robin, and Lori for helping us put everything together, and of course, Brian and Pavan.